Well, first of all, hello everyone. I'm Francois Orange, and first of all, I wanted to thank Rutching and Tom for giving me this wonderful opportunity to give this talk to all of you. So in this talk, I'm going to talk to you about array tomography, and I'm going to talk to you about giant cells in root nodes nematodes. But first, a little word about the place I'm working. I'm working at the Université Côte d'Azur, which is located in Nice, in France, on the French Riviera. So you can see between the sea and the mountains where our facility is. We have the chance of working in a World Heritage Site university, and actually the picture I'm showing to you is not just for the show. Our facility is actually behind this building there. And so the Centre Commun de Microscopie Appliquée is the electron microscopy facility of the Université Côte d'Azur. So here is our team. So we're 5 people among the team. So it's a picture where only 4 of us because Andrea just joined us recently. We didn't took another picture yet. So we have more than 35 years of expertise in electron microscopy. We're open to everyone, laboratories and private companies. And we are multidisciplinary. We work mostly on life sciences, but we also provide local solutions for material sciences. And overall, you can see our equipment on the right. We have 2 scanning electron microscopes. We have 1 transmission electron microscope. None of them are state-of-the-art electron microscope, but they are a good microscope, and we're doing our best to obtain some good images and data out of it. And I think we're doing a pretty good job. And overall, we are what I like to call a human-sized facility. A quick word about myself. So you had the introduction on the website, but I started years ago with a PhD in astrobiology in Orléans in France. So I used to work on the experimental fossilization of extremophilic microorganisms. I tried to obtain microfossils of extremophilic bacteria, like the one you can find in the hot spring of the Yellowstone National Park, the one you can see on the top right. After a few postdocs in astrobiology, I switched in 2011 to electron microscopy. Then, after another few postdocs in Puerto Rico and in France, I got a position as an engineer in Nice, where I've been working since 2014. And there, on a facility, I'm in charge of the scanning electron microscopy. And I like to do also some methodological development in everything that is related to 3D, like array tomography or photogrammetry applied to SEM. So in this presentation, I'm going to talk to you about a paper which we published last year in New Phytologist. It was a collaboration with people from the INRAE. It's Agronomy Institute in Sophia Antipolis, a few kilometers away from Nice, and mainly with Janice de Almeida Engler, which is from the Brazilian Academy of Science and has been working for possibly more than 30 years on root nodes, nematodes, and galls. So in this presentation, I would talk a little bit about infection of plants by root nodes nematode, and later on array tomography. If you know nothing about root node nematode, don't run away. I just have a few slides on it. Then I will switch to a full technical description of array tomography. So a quick word about the background. While we have some pathogens which induce plant-damaging tumors in plants, which cause global crop losses. For example, on the top right, you have a picture of galls, some tumors induced by root nodes nematode, so small worm that penetrate the roots and will feed with what they find within these roots. Due to molecules secreted by these nematodes, you will have some problems created on the mitosis, and that will induce also a high metabolic activity, which will induce a proliferation of organelles and a large nucleus. The result will be giant cells within these galls with multinucleate hypertrophied feeding cells. You can see a picture on the top right where you have, just here, you can see the nematode, and all these are the giant cells. We'll see a bigger picture of this. And so just a few examples of these giant cells induced by nematode in tobacco, soybean, or Arabidopsis. And what we got within this are a nucleus, which is multilobate structure. And this is something which has been characterized already by light microscopy and transmission electron microscopy. But to know more about the structure of this nucleus, we need for 3D data at a submicrometric scale. And that's where we need to do some volume EM. So the next slide will be pretty much basic, but there are for the possibly some people among you who may not be familiar with all the 3D technique, why and how we are doing this. So there will be some basic stuff, some stuff that's been already said in the previous presentation. But the basic of this is that with the TEM, we have the best resolution, but we're doing observation and, on a 2-dimension, and it can create difficulties for the understanding or the morphology of the observed structure. I give an example, for example, of a macrophage. If you have a section along the red line, it might give the feeling that you have 2 nuclei. Well, actually, it's only 1. So we need for 3-dimension, but how? You can observe within the thickness of a section, that's electron tomography, or you can observe, like a confocal microscope is doing, serial section, and then you will get into volume electron microscopy. And the last decades, I've seen major progress in volume electron microscopy. The previous presentation was an example. And they all allow the 3D visualization of biological ultrastructure at a very high resolution and on large volumes, up to 1 cubic millimeters or maybe more. So these are, like on the, I'm sorry, it's a repeat of the previous presentation, but these are the techniques available for the volume electron microscopy. On the left, you have the historical one, like electron tomography, but only suitable for small structure. You also have serial section, TEM, where you have a nice resolution, but the collection of section will be tricky and the overall grid will be fragile. On the right, the other and most recent volume EM technique, you will have the serial section, SEM, the array tomography, where you still have the tricky part of the collection of section, but you can collect more on a hard support. Serial block face and focused ion beam, you will put your resin block with your sample embedded in it within the chamber or the microscope and mill or cut it with automation, and then you can go into a very large volume of data, but they are destructive. To sum up, I like this sentence from the paper from Bingban and Bock from 2012. Actually, there is no best volume EM method. Each method has its pros and cons. Many years ago, when I got into electron microscopy, I was amazed at conference to see all these beautiful 3D animation. I was in awe, and I wanted to do the same. But there were a few problems. Is that you need time, you need money, you need possibly a dedicated instrument. And so possibly it's a problem feeling shared by other people. People might be reluctant to get involved in volume EM since it's often considered highly complex and time-consuming approach with the acquisition, the segmentation, reconstruction time, and also requiring expensive equipment. But among the techniques I told you about, you have array tomography, which for me is an excellent entry point in volume EM. If you collect just a few dozen of filters in section on a support, it's enough to get valuable data within a reasonable amount of time and cost. It would take you only a few hours to acquire and to process the images. And also you can do this with equipment, which is commonly found in electron microscopy facilities. So you don't need a dedicated instrument, which means that it is an accessible technique. And for me, it's very important. You will have cost efficiency. Array tomography allows flexible protocols, possibility to do correlative and light electron microscopy, and also section can be kept and observed again. At the bottom of this slide, I just mentioned 2 papers. The first one, all of them by Krystyna Micheva, which created the word array tomography back in 2007, the paper on the left, to describe the technique. And Krystyna Micheva and colleagues Jemima Burden and Martina Schefferer published a paper 2 years ago, still on array tomography, but just to mention once again that array tomography is still relevant. You have a website, which is arraytomography.org, where you will find, if you're interested, a lot of resources on it, protocols, tricks, and everything. A very useful website. So still on the basic of the technique, well, what we're doing is that on array tomography, we collect the section on a flat surface, which can be a silicon wafer, a glass slide, a tape, like we've seen on the previous presentation, and you observe it in the scanning electron microscope. So basically, you may be wondering, so you're doing TEM with an SEM? Yes, absolutely, we do. And how does it work? Well, the contrasting agent used for TEM, we have heavy metals like osmium, lead, and uranium. This heavy metal, when observed in the SEM using the backscatter electron, which work with a chemical contrast, this area stain will appear brighter. So what you will have, instead of a TEM image on the left, you will have a SEM image, and all the area stained with this heavy metal will appear brighter. But if you invert the contrast, you will have the equivalent of a TEM image. So you can collect, as I said, a higher number of several sections. You can keep them. The problem, you won't have the resolution of the TEM, but you won't be that far, actually. On the next slide, I will describe my own setup for the array tomography. And also, I will assess at several points that all this is accessible, because everything is, all the steps are quite simple. The first one is the sample preparation. In that case, in that study, we used our routine TEM protocol with the glutathione fixation, osmium post-fixation, dehydration upon resin. No additional contrast for this study. Then, for the section collection for array tomography, well, it's a little bit more. It's not that complicated, actually. I want to assess that if you know how to use an ultramicrotome, you can do array tomography. You still need to take a few precautions, like you will need to have a very well trimmed block face. But in that case, you see the entire setup. I use a diamond knife, which is my common diamond knife. It's not even a big one. I just put a glass slide on it, made hydrophilic. And what I do, I collect a ribbon. And once the ribbon is obtained, I detach it, and I will attach it to the water-air interface on the glass slide there. With a peristaltic pump, then I will decrease the water level, let it to dry. That's it. So it's something which is, not that complicated, actually. Further on, for the staining and coating, nothing new there. The same procedure for post-staining as for the TEM grid with the uranyl acetate and lead citrate. The glass slide were mounted on SEM stub, some carbon coating, and we go into the microscope. Our scanning electron microscope, most of the data we show within this presentation were obtained with SEM equipped with a tungsten filament with the YAG backscatter electron detector, which means that it's not the best SEM to do AT. It's pretty much an all-around SEM that we have in our facility, but it works nevertheless. You can acquire AT data, volume EM data with this microscope. The downside is that all the acquisition takes time and have to be done manually, but it works. And on the right is the kind of images that I obtained with this microscope. The last step of this workflow will be the image processing, where I've used the iMod software, which is an open source software for the alignment, the segmentation, and the 3D modelization. iMod allows to do all of this. The alignment tool, because you will have images from successive slides will not be perfectly aligned. You need to do the alignment and to correct eventual distortion. And there is a very powerful tool within iMod for doing that. The thing is that we'll have, like the picture we see here, two successive images. One will be displayed in red, the other one in green. You need to overlay them exactly. You can do that automatically, but you can correct it afterwards. And if it appears black, then it's done. Then, still with the same software, the segmentation, which can be the rate-limiting step of the entire process. And eventually, you generate the 3D model. So in summary, in our case, we use a routine TEM protocol, an old ultramicrotome. We collect section on glass slides. We use an old carbon coater, a scanning electron microscope with a tungsten filament, and process it with an open source software, which means that the cost for implementation of volume EM in our facility was zero. So, of course, as I said, all this step has to be done manually, and we will need, of course, automation in the future. But to get into volume EM, It cost us nothing. Now we'll go back to the study of the giant cell in the root produced by root loss nematode. So basically, on top left, here are the glass slide on which I had my ribbon of section on it. I've collected around 400 sections with a thickness of 100 nanometers. I didn't go straight into the full imaging of this section. I made further survey to check if it was relevant to image all the section. That's what you see on the bottom left. I made one image of the entire gal every 10 sections. And on this, I noticed that one of these giant cells here had a nucleus which showed a concentrical structure and actually looked very good. That was exactly what I was looking for. And it proved to be actually a gold mine eventually. I will show you this in the next slides. So when I saw that, I was like, okay, let's go for this. Let's go for the entire imaging of the 400 section. Sometime later, I was able to produce a stack of images and an aligned stack. That's what you're seeing here. It's aligned with the IML software. So basically, I had a bit more than 400 sections collected. Some of them were removed or missing. The section removed were the one where you had some folds or dirts, the usual artifact and problems you can have with just usual ultrasonic sectioning. Overall, we had maybe around 12% loss, but actually it was not an issue for the segmentation. You notice that once we go further in the stack, most of the missing sections were from the last third of the stack. That's the more and more we're sectioning, the more and more that knife was getting dirty, and we had problems of folds or dirts with the section. And sometime later, actually quite a lot of time, here is the reconstructed 3D model that I got from the nucleus of a giant cell. It will appear shortly. Here we go. What we've noticed is that we know that you had many lobes and stuff like that, but actually we saw that it was actually one single object. For the sake of clarity, we subdivided in a subcluster in yellow and green. In red were the lobes not connected to the other part, but they were in the limit of the stack. So possibly if you go further within the section, you could have seen some connection. And each subcluster were made of multiple nuclear lobes. Just a quick slide for the comparison of the size of this nucleus in a giant cell and the nucleus in a normal cell, like the small one you can see in the vicinity of this giant cell. You can see there's a lot of difference in the size and in the morphology. So this cluster were made of nuclear lobes. You had simple nuclear lobes, is one of them. You had in green one more complex nuclear lobes. Each of them has several nucleoli in it. But within the middle of the stack, we find very much in the middle one lobe which looked different. We call it the central area because it was pretty much in the middle of the stack. But most importantly, the structure was different from all the other ones. It looked like wrapped layers of nuclear material, and it contained only one nucleolus. When doing the segmentation, when I was checking if I hadn't forgotten anything, I noticed within the images some weird stuff, like the one where you can see if you can see the pointer there, small tiny stuff, tiny structure that I could follow from one section to another, and go on from many sections like this. And I was like, what it is? And it turns out to be fine tubular structure coming out of the nuclear lobes. With my co-author, we decided to call it nuclear tubes. These are tubes which have a diameter of like 150 to 200 nanometers, but which has a length up to several tenths of micrometers. It's very long. And you can see easily from this animation how this is the kind of stuff that you could have missed on the TM observation on a single ultra-thin section. With the fact that you have serial section and that you have the resolution of an electron microscope, it's the only way to be able to identify this structure. So volume EM was absolutely necessary to identify these structures And in addition, the fact that with array tomography, you can keep your section and go back to it to do more images at a higher magnification was very important as well. So these tubes, so we done some higher magnification picture on this. You can see that you had a double membrane. It was clear that it was coming out nuclear lobes. Like on this animation, you can see one coming out of the nuclear lobe here. One weird thing is that all these nuclear tubes were rejoining in two specific areas. They were converging in two specific points. We call them nuclear converging points. And this one was also one of the discoveries of this study. The thing is that we don't know what's happening there because we missed the resolution on these converging points. I tried to do the highest resolution possible with the microscopes we have here, but actually still don't know what's happening there. Is there a fusion of the nuclear tubes? Is it a knot? We don't know. So overall, you can see that these nuclear tubes just create a network within the nuclear cluster. You have all these white line there. You can see the network of the nuclear tubes. So they have in summary, they have a double membrane, or likely extension of the nuclei. They can see sometimes as pairs or triplets. And also, important thing, there is no intermediate forms, no sprouting that we detected. And the next question is, what's the use? What's the role of these nuclear tubes? So we have no answer yet. It still is an open question, but some possibilities, some hypotheses could be, it could be used for the optimization of the metabolic activity of these giant cells. It could be used for nuclear communication or for transfer. What we could see if we go into this nucleus is that also this nuclear tube, some nuclear tubes connect the two converging points between them. One converging point here, another one here, and you have a few nuclear tubes connecting the 2 points. One of the things we've discovered within this nucleus, we also find some thick membraneless tubes crossing the nucleus and exiting with no link with the nuclear lobes. They was just there crossing. They had a beginning, they had an end, and they were surrounded, like you can see on these images, by a densely stained sheath. You can see on the animation there, 2 of them within the nucleus. Their role is still an open question as well. One possibility is that it could be remnants of the feeding tubes of the nematode. The feeding tubes would be like a needle that the nematode would put into a giant cell to feed himself. Possibly this tube could be a remnant of this, but it remains to be proven. When trying to understand what we saw, we try to find similarities in other systems. And I give you 2 examples there. We know that, for example, in plants, you have the chloroplast and the mitochondria, which can communicate via tubes. In irradiated cancer cell, you will have also structure which looks like the nucleus we found with some multilobate structure with some like tubes connecting all of this and some converging point as well. So this could be similarity and gives hypothesis to understand what we saw exactly. Some concluding remarks. So on the part of the study of the nematodes and the guards, so this 3D visualization revealed the presence of nuclear tubes, which could be the first report on nuclear communication. The discovery of these nuclear tubes, it's a unique component of this nuclear cluster. And this could be exploited to develop strategy to control root nodes nematode. Like, for example, if you can target proteins that helps to build up this nuclear tube, for example. And back on the array tomography from a technical point of view, the study also illustrates that it is possible to apply array tomography to roots affected by nematode using basic equipment found in most EM facility. But as I said a little bit, it took a lot of time to obtain this model. So for further study, we will need automation for future volume EM study. And so our plan at the moment in our facility is to get a new SEM where we could do some automation or this brand new fancy thing that we could do nowadays on state-of-the-art microscope. So that's the end of my presentation. I would like to thank you very much for your attention.
Thank you, Francois. There's already several questions in the chat. So let's start. First, Melissa asked, how do you handle charging of the glass slides while imaging?
Well, to be honest, I had no charging on my glass slide. I handled it with the. I will show you the slide once again. I do some carbon coating. So you see there we go. On the SEM stub, I stick the glass slide with some carbon tape at the bottom, on the top, carbon coated. Never had a real issue with charging effects on this.
Okay. The next question is, what are your tools for staining the sections on the glass slides?
Pretty much the same as the staining of the TEM grid. Right. For the TEM grid, I would put a drop of, let's say, staining agent. There I will put the drop on the glass slide. Then I will do the same time for the staining as for TEM grid. Then I will do the rinsing with some water on it, and that's it. I haven't invented nothing for this. I just took for the sake of simplicity. It's the same procedure for TEM grid.
Okay, so two more questions. Melissa asked again, have you tried doing stack alignments in Fuji using SIFT or something similar instead of iMod?
I haven't. I will go back to the alignment slide. I haven't because I went into the Midas module from iMod, which I use for the alignment, and I find it very powerful, especially for array tomography. I think, correct me if I'm wrong, is that I don't know if the SIFT element method from Fiji can do. It's just only a cross correlation and just a translation, possibly a rotation of this. But I know that Midas is very powerful because it takes into account the distortion, the stretching of the section, which will happen in the case of array tomography. You will have some stretching, and you need to apply some stretching of your images for the alignment. Otherwise, you will have a stack which is pretty much kind of jumpy and not very accurate. And for this, Midas is very powerful. In my plan, still possibly in the years to come, I will try to find a software which will do the segmentation more or less automatically. But I think I will stick to the alignment tool of iMod because it's very, very powerful and quite easy to learn and to master.
Okay, you have
actually in complement for this alignment, one day I decided I decided to learn how to use this tool. I decided to keep for myself and then half a day to learn how to use this tool. 10 minutes after, I had already aligned my stack because it was really simple.
Good to know. Yeah, so Steph asked a question about the nuclear tubes. Are they found in which stage of the nematodes? Adult or larvae? She also like to know they are in plant cells. Are they in plant cells?
They are in the plant cells. All the things that I've showed to you are in the plants. The nematode is not here. You can see this is a giant cell from the plants which have been produced as a result of the presence of the nematode, but this is a plant cell, and so the nuclear tube will happen with this nucleus there.
Okay, so there's some more comments. There's one more question, two more questions are coming in. How do you avoid wrinkles on the sections?
The wrinkle of the section. Well, so this is certainly a place where I have room for improvement because, well, you will have wrinkles. I would say the same tricks that you can use for common ultra thin sectioning can be applied for this. I know I haven't tried. You can play with some chloroform vapor to stretch a little bit the section and avoid the wrinkling. Some people put a few drops of ethanol in the boat of the diamond knife. So there are many tricks which I think can apply to this. I haven't used a specific thing to avoid wrinkles, so I had wrinkles. One thing also which could prevent wrinkle would be the slow process of deposition of the ribbon on the glass slide when you decrease the level of the water. It will kind of very slowly and very nicely the section will be deposed on the glass slide, and possibly you can have a little bit of stretching there which can remove a few of the wrinkles. But I can tell you I had wrinkles in there, I had folds, I had to remove sections within my stack because this wrinkle could create artifacts and problems for the alignment, so I removed them. But I could keep the information within this section if I had a doubt when I was doing the segmentation.
Okay, so she has a follow-up question about the glass slide. Did you treat it with plasma cleaning before you use them?
Glow discharge, yeah, beforehand. I think
Okay.
it's necessary to have a very nice hair water interface beforehand. So you do this just before doing your sectioning, and that will allow you to really, you can see a little bit here and there, have a really nice hair water interface. Without it, that would be kind of complicated, to be honest.